So, what about Neanderthal DNA? Hey guys, I'm Brian Osborne with Answers in Genesis here with Dr. Nathaniel Jeanson, also with Answers in Genesis, doing a whole series on his brand new book called Traced Humanity's New Rosetta Stone. We're calling it Human DNA's Big Surprise. And last time we were talking about how really Dr. Nathaniel's research, Dr. Jeanson's research, has done a phenomenal job of doing actual science. It's reached that gold standard. It's made predictions. Those predictions have been fulfilled, and there are new predictions being made, and we look forward to seeing how those are dealt with later on in the future. Lord willing, those are fulfilled as well. We keep progressing in this realm of science and seeing science confirm the Bible. And so we've seen a lot that are already excited about the future, but a big question that looms kind of over all of this as we talk about mutational rates and the father son Y chromosomes, all these sorts of things, showing that with the human growth chart, human growth over time. But how does that relate to Neanderthals? Where do they fit in on all of this? And can really this research done in the book Traced accommodate and fit what we see with science, fit with what we see with Neanderthals, and then explain them as well? That's kind of what the session is going to be all about. I'm excited about it. Dr. Jeanson, all yours.
Thank you very much, Brian. And just for those who are joining us live and not binge watching this, we're in part 12 of a larger series that you can find on the Answers in Genesis YouTube channel dealing with this book, as Brian mentioned. We're in step 2, basically, of a little mini series dealing with the question, how do we know this is true? If you've watched the earlier episodes, you know we've gone through some of the main content of the book that deals with human history and the history of peoples for Africa and Europe and the Middle East and South Asia and East Asia and Native Americans and all that. How do we know these conclusions are valid? One of the main concerns I anticipate would be, well, you're basing this within a young Earth framework. This does not agree with the mainstream timescale. And in fact, if you looked at the earlier episodes, I made a very strong statement saying this book represents one of the strongest arguments in print for the recent origin of humanity, going back just 4,500 years to Noah and his sons. Well, how do we know any of this is true? That's a valid question to ask. And if you've not read the book, or maybe someone introduced you to this book and you say, well, I don't know if I buy this. These are creationists. How do we know this is true? I would go back to what the evolutionists have said. I gave you a few more quotes in the previous episode. I refer you back there for more of this and some of the background to it. But there's a strong philosophical and long-standing debate background to why I'm going the direction that I do. And it has to do with scientific testability. What have the evolutionists said is the gold standard of science? What defines science versus pseudoscience? The ability to make and fulfill testable predictions. Can we, in theory, disprove what I'm saying? And the answer that I tried to begin advancing in the previous episode, and that I will try to continue advancing in this in subsequent episodes, a point I'll come back to again and again, is that I would say we know this is true because it's part of a historical progression of discoveries in which successive, testable young Earth creation predictions keep working. We began to look at step 1, some of the initial findings with the Y chromosome. We're going to, by the end of the series, have looked at an entire decade of young Earth creation genetic results. Again, the age of the Earth debate involves multiple disciplines. I'm a biologist focusing on genetics. I'm going to give you the answers from the field in which I have expertise. I can't speak to you as an expert on geology, paleontology. I refer you to the works of others, my colleagues who've done that sort of research. This is for the field that we're looking at in Traced. And for the general questions of human genetic origins, how do we know that this timescale, this model that I've laid out is true? How did we get to the point where we can look at this tree? And again, the big picture is we're looking at a DNA-based generation by generation family tree for global humanity based on the male inherited DNA, based on the Y chromosome. How did we get to the point where we could read history off of this tree and do so in a manner that treats the length of human history as just a few thousand years long? How did we get there? Last time we looked at some of the evidence, the first lines of evidence, looking at father-son mutation rates. And by the way, let me just emphasize here again. I'm going to give you a semi-chronological narrative. That's the main point of this little mini-series, is to lay out for you, here's the sequence of historical events, papers, predictions that were made over time. I've, in a sense, jumped into the middle of the story. I've said, let's begin by looking specifically at the sequence of Y chromosome-based discoveries, because that's the focus of the book Traced. But there's more to the story. There's even more backstory dealing with other DNA compartments for humans that we'll get to by the end of this little mini series. So we began with the Y chromosome, going back, some of this research started for me in 2017, 18, and then one of the first papers on this in 2019. And that paper summarized some of the data in the mainstream literature looking at the rate at which fathers make mistakes in the Y chromosome as their sperm stem cells copy it and then eventually pass it on to their offspring, male offspring. We looked at the history within the mainstream literature of the results that were published along these lines, the measurements, the experimental measurements of fathers versus sons, brothers versus brother sequence, where they have a known Y chromosome father ancestor. One of the early studies in 2009, 2 Chinese men, common ancestor back in the 1800s. Again, some of this review is for those who are watching live. If you're binge watching, appreciate your patience. We talked about, and again, I'll refer you back to it because it's a long discussion, the difference between high coverage and low coverage. That's the technical term for quality of the data. And it turns out that the 2 high quality studies yielded and discovered a very fast mutation rate. If you think about the Y chromosome, the male inherited DNA as a clock, this implies that the clock ticks quickly. So if you were to work your way backwards in time and dial the clock back to 0, you could do it in just 4,500 years, implying that our common male ancestor lived in the recent past, not hundreds of thousands of years ago. And shockingly, the mainstream community responded to their own data by literally filtering it out. In the case of this 2015 study, they say that explicitly in the paper. This rate, which was 10 times higher than the rate based on the low quality data, this is about 3 mistakes per generation. This is 0.3 mistakes per generation. 0.3 letters mistakes per generation. 3 letters that are in error per generation. This study literally filtered stuff out. In this particular study, they focused on fairly exhaustively the rate at which DNA changes, not just the Y chromosome, but other types of DNA change from parents to offspring. And they left out any sort of explicit statement on the Y chromosome. I was able to extract from some of their raw data that did slip through a mutation rate. And again, it was high, consistent with this other high quality study. So long story short, it does look like father-son mutation rate is indeed fast. And this would imply that we can, with a fast ticking of the clock, you can dial that clock back to 0 in just a few thousand years. Well, the gold standard of science is making testable predictions. And oftentimes, predictions arise out of objections. Some of the objections come from my ideological opponents. Sometimes I just have to think through the data myself and say, okay, I got to be my own worst critic. What are the holes in my argument? What are the assumptions that I'm making? One of the assumptions I'm making, if I'm dialing the clock back to 0 with this father-son mutation rate, I'm assuming that this rate we measure now has been consistent through time. That's an assumption that creationists have criticized evolutionists for making in other fields of science. Common assumption in geology works its way into astronomy. If you measure the rate at which the Colorado River is currently eroding the Grand Canyon, it's fairly slow. If you assume that rate has been consistent through time, going backwards in time, you get a very ancient age for the origin of the Grand Canyon. And creation scientists have said that's a big assumption. You're basically ignoring the possibility of a global flood. That's been the debate in that field. I bring that up because now it gets fresh legs, but in a sense, a role reversal in the field of genetics. One of the ways to say this is, well, how do you know natural selection didn't play a role? Perhaps, yes, mutations occur fast now, but over time, natural selection will eliminate some of those. And so effectively, the clock ticks slower. There's another objection to all this. One of the things you'll see in the book, and that this whole research is based on, is looking at the DNA of living people. This tree that I just showed you, that the book uses as one of its primary foci to read off human history. This is based on comparing the Y chromosomes of living men. There aren't that many differences among us, so it's fairly easy to line up our Y chromosome sequences, count off the number of differences, and then plug in these numbers into a program that'll reconstruct a family tree. What's not included is the published DNA sequences from fossils like Neanderthals. It's one of the things we mentioned at the top of this episode. So what do you do with that? Isn't that a potential objection to what you're saying? All these things that I'm discussing are testable hypotheses. The hallmark of science is the ability to make testable predictions. So what I just showed in the previous episode about the fast rate of mutation that implies a recent origin for humanity, what testable predictions does that view make? Again, it excludes ancient DNA. It makes assumptions about the rate of change and its constancy. What predictions does that make? And conversely, when we think about the objections to that view, that conclusion, what predictions do the objections make? So just to summarize then, I mentioned that I didn't include ancient DNA in that family tree. So if we return to one of the diagrams from the previous episode where I talked about a fast rate of mutation implies that over hundreds of thousands of years, an enormous amount of mutations would occur, far more than what we see. Well, what if what we see, this bar right here, what if it includes ancient DNA? The thought is, well, this bar will go up, and now the data will be in alignment with evolution. Or to put it in the context of this graph, where I say that this fast rate of father-son mutation applied over 4,500 years captures a lot of the DNA diversity that we see. Not so fast, an objector might say. If you include ancient DNA, now these bars that you try to capture go way up. And this discounts your conclusions. You cannot dial the clock back to 0 in 4,500 years. It takes longer than that. So your ideas can't possibly be true. So what predictions do these views make? And so just a diagram for you what I'm talking about. Father-son mutation rate is measured. How do you know that this rate we can consistently apply going backwards in time? And sorry, before I get to the predictions here, just to show you this is in fact something that critics have published as an objection. Dan Cardinal has specifically raised this point. You can see his video, the book review traced on YouTube, where he says, I'm conflating genealogy and phylogeny. Basically, I'm taking a short-term rate and erroneously applying it backwards in time. The technical terms for this are substitution rate versus mutation rate. He says the mutation rate is the father-son, the substitution rate is the long-term rate. I'm conflating the two. This is just a fundamental error, textbook error. Therefore, my conclusions are invalid. And he also raises the point about, see, I excluded Neanderthals. So I'm not just inventing these objections. You can actually find these out there in response to this book. We'll have more specific objections or more specific responses to some of these explicit statements from the critics in later episodes. The purpose of this little mini-series is to lay out for you the history. But this is some of the background to it. The key element, again, is testable predictions. One of the first predictions that I want to show you comes from a concept, if you've, again, watched this series basically from beginning to end so far, deals with a concept that we already touched on and that I also touch on in the book. And this is the concept of how family trees record changes in population size. For Christians, creationists who are likely familiar with the biblical anthropology and the origins in the Old Testament of the nation of Israel, you're familiar with Abraham, Isaac, Jacob, and then, of course, Jacob's 12 sons. Talking about sons because we're talking about the Y chromosome. If you were to draw out the family tree of Jacob and his 12 sons, you'd have one branch coming from Jacob and then expanding into 12 branches, leading to the 12 sons, which became the ancestors of the 12 tribes of Israel. Take away the names, you can see a fundamental, simple concept that the number of branches increases from 1 to 12. Family trees record fundamental information about changes in population size, rises and falls. This is true for nuclear families. It's true around the globe. So, this leads us then to a testable prediction. We know from archaeology and historical records, i.e., we know independent of genetics, the general shape of human population growth throughout history. I've got it here in this diagram. Negative numbers represent the BC era, positive numbers the AD era. I've got year 0, even though it doesn't exist because it's easier to graph this way. And what you can see about the general shape, on this graph here, you have billions of people, 1 billion, 2 billion, 3 billion, that human history has had a fairly flat or modest rate of growth. If we were to zoom in here, you could see some of the nuance, the ups and downs throughout human history. But perhaps the most visually striking aspect of human population growth is this sudden spike upwards in the last few centuries. In 1400 AD, it's estimated that the human population size was just 350 million people total, the entire planet. Now, of course, it's approaching 8 billion. And once again, I want to emphasize this is what's known from archaeology and historical records independent of genetics. In fact, I'm taking this from a source from the 1970s, before anyone had access to the Y chromosome and the complete human DNA sequence. So here's a data set by which we can test a prediction. So I said, step one, we measured father-son mutation rates. In a sense, if you look at this family tree, we're looking at the very tips of these branches. Time moves from top to bottom of this family tree. And I'm saying if I measure fathers and sons at the very tips of these branches, and it seems to be that the rate of change is fast, and then I assume that rate has been constant all throughout the tree, you get a recent origin for humanity. Well, how do we know that this assumption is true? What predictions does it make? Family trees record the history of human population growth. If you have a representative sample of men around the globe, you reconstruct their family tree and you read off the population history. Does it match this? That's how you test it. So one of the predictions from what I said in the last episode is that if this rate is indeed fast and it's been consistent throughout human history, you should be able to see a history of human population growth that looks like this hockey stick shape. In other words, if there's mutations happening every single generation and new branches are being laid down every single generation, you should be able to record this history, hockey stick shape, in a generation by generation by generation manner. So in 2019, one of the companion papers, I'll give you the reference here in a moment, did just that. It tested that prediction. That's what I put in the title. You'll see it here in a moment. These two black lines represent the range, if you can see them here, thin black lines represent the range of the estimates for human population history based on archaeology and historical records, not genetics. That's the test data set. I'm going to test in blue the implied history of human population growth against it. So the two blue lines represent the range of estimated history of human population growth. Looking at this Y chromosome tree, saying, okay, if Noah's here, and this is just 4,500 years of history, and we say new branches every generation, I lay out the details of the methods in the paper. What sort of population history do we get? And we get a hockey stick shape. And the match between the blue and the black is about 94%. And you can look through the details of the paper. I tested all sorts of different positions to say, does the overall history from genetics match the history from archaeology and historical records? And the answer is yes, depending on where you put Noah in the tree. There are some positions that are very poor match, some that are much better. So this is a direct test of a prediction and a direct answer to an objection. And it works very well. That assumption works very well. There was also a way for me to use the same type of analysis to test the ancient DNA hypothesis. So let me just lay it out for you in graphical form here in a moment over the next few slides. Taking data from a paper 2020. So this comes out after my 2019 paper was published, but there are other ways I tried to estimate how this all works out. But this is a nice visual for us to look at this. They draw the family tree for humanity in an evolutionary based way where this branch, if you extended it backward in time, would take you back to the common ancestor that humans supposedly had with chimpanzees. And here you have a Denisovan. This represents DNA differences and leading up to Denisovan individuals, Neanderthal individuals. Here's the very long branch, A00. To help you connect these visuals, I just showed you this diagram, which is based on the DNA of living men. The evolutionists are basically saying this whole diagram is captured right down here. So, I mean, you have to reorient the tree slightly because I've got A right here when in fact A comes off here. But the point is, to get back to these sequences of Neanderthals and Denisovans, you have to go back a long way. There's a lot of mutations to get you back there. That's, again, part of the reason the evolutionists would object and say, in fact, they might even go so far to say the creationists are fitting facts to conclusions. Neanderthals, the fact that there's so much DNA diversity, there's more DNA differences between Neanderthals and us than between any two living people today. The reason this isn't entraced is because it would be a contradiction to those conclusions, and creationists ignore data that contradicts, and they include data that fits. It's not science. In 2019, I actually tested this hypothesis with this tree orientation. I said, okay, what if it is true that if we say Noah is back here or back here, that Neanderthals are his post-flood descendants of Noah after the flood, and you have to reorient the tree and compress a lot of history. you can test that hypothesis. How does the branching structure, the history of human population growth, look? That was tested in this paper as well. Once again, the two black lines represent the history of human population growth, archaeology and genetics. Archaeology and human history, not genetics. The blue lines represent the history of human population growth as inferred from the human DNA tree, Y chromosome tree, when you include something that approximates a Neanderthal sequence. And the result is a very poor match. This is my experimental rejection of including Neanderthal sequences. So what's the explanation? What's going on with Neanderthal sequence? It might be unreliable. It might be something else. Whatever is going on, if you treat Neanderthal DNA sequences as reliable and that they represent a group of post-flood individuals from Noah who mutated their DNA at a constant rate, you do not get an accurate reproduction of human history. That's an experimental finding. So let me clarify. If you base a human family tree just on the DNA of living people, and you get their connections from their DNA differences, and then you look at the implied history of human population growth, you get a very good match. Strong scientific argument in favor of taking that approach. If you say, what if we test an alternative approach where we include fossil DNA sequence, assume it's reliable, constant rate of mutation, you get a very poor match. Why is this match so bad? That's the question. And why is this match so good? Those are the predictions I was testing in this paper, companion paper. Again, we're still in 2019. The point being, you can very successfully capture the history of human population growth when you do so from a tree based on the DNA from living people, and you have an overall time frame of just 4,500 years. So there's all sorts of implications that flow from this. If the mainstream timescale is correct, why does this timescale recapitulate human history so well? Why is the father-son mutation rate fast, in line with the expectations of the 4,500-year timescale? And why is the implied human history, the history of human population growth captured so well by the same timescale and same set of assumptions? And why, when we include ancient DNA, does the match drop so low? The conclusion I made from this was that there's something amiss with this sequence. It doesn't work very well scientifically. And so I've been excluding it and excluded it from the book. This isn't the end of the story. There's more predictions that flow from even these two steps. But again, the focus is making predictions, because the most important feature of scientific hypotheses is that they are testable. The data that I just showed you was matching the global history of human population growth with a reconstruction based on a global sample of humanity, their DNA. It predicts then you should be able to do a similar sort of analysis and find a similar sort of match based on regional samples of humans. And we've got, of course, from archaeology and human history, regional histories of human population growth. Let me give you one example. It's now well known in mainstream archaeology that there once existed a massive number of Native Americans on the eve of the arrival of Columbus and his colleagues. And that within a few centuries, in some cases just a few decades after the arrival of Europeans, almost 90% of those people disappeared. One of the most tragic eras of human history. So to give you a comparison, in 1400, one estimate of the population size for Europe, all of Europe, is about 60 million people. One middle-of-the-road estimate for the total amount of Native Americans in the Americas in 1491 is 50 to 60 million people. And then by 1800, nearly 90% of them gone. You can read more about this in this book, 1491. Charles Mann is a science writer, not a creationist by any stretch of the imagination, assumes the old Earth timescale, but he narrates some of the revolution that has occurred in mainstream science in the last half century along these lines of population sizes. Go back 50, 60, 70 years, and the consensus view was that there were hardly any people here, living sort of in harmony with the environment, small bands. All that's undergoing a dramatic change in thinking. And it's now viewed that there were massive numbers of people and that population collapsed. This is in the back of my mind when I was starting to work on Y chromosome research. I live in Kentucky. This was in our local library, and was the whole history of the pre-Columbian Americas was a big black box for me for much of my life. We never learned about it, any details hardly, in high school when I took history. And I had long wondered what was actually going on here. And so to find this book in our local library, devoured it eagerly. And I had this concept then in my head when I was working on the Y chromosome tree. So where is the smoking gun of this? Here we have a regional and, in fact, sort of a short-term population history for the Americas. Lots of people, then boom, big collapse for about 300 years. And I should add here, about the 18 or 1900s, a recovery. The exact date depends on where you are in the Americas. For, let's say, the Navajos and some of the Western nations, it's sort of late 1800s where you finally begin to see that tide shifting. And as the Europeans push westward and establish new states and such, and it leads to these inevitable conflicts with the Native Americans and wars and destruction and such. Once all that finally gets resolved, if resolved is even an appropriate term, they're forcibly removed to reservations, and the populations begin to recover. So that's fairly late 1800s. Okay, well, let me give you a theoretical diagram of how a family tree might look in real time if you were watching Native American family trees with a time machine and could watch how the populations grew, collapsed, and recovered. Here's 1200, before Europeans arrive, and I'm hypothetically diagramming here, an increasing number of branches that form with time as the population's growing, up to maybe 50, 60 million people. 1492 right here, so time moves from left to right. 1492 arrives, Columbus arrives, here's this vertical dashed line, and within a few centuries, boom, a whole bunch of branches are gone. Villages here, disease arrives, some of them enslaved, killed in wars. And you might note here that I've got 10 branches and have killed off 9 of them, representing about a 90% collapse. And then just a few stragglers survive, and then the branches starting again about the 1800s. That's how it would look in real time. We don't have access to that if you take the DNA from living people. Let's say you took DNA from living Native Americans. What would be the signature in their family tree, the smoking gun of this history? All these people are gone. They've got no descendants to this day. You'd see these branches coming off because there's no descendants. Flatlining would be a good summary of the smoking gun. And there's a deliberate double meaning there. Of course, we use it in the medical field to talk about people dying. The signature of a massive die-off is a flat line. If you look at, actually, if I could had more time to narrate for you how I actually went about coming to the conclusions that I did, the story really starts back in 2017 when me and a colleague, Rob Carter from Creation Ministries International, were working on trying to use the Y chromosome and the male inherited DNA and the female inherited DNA, the mitochondrial DNA, to see if we could find the genetic echo of the ugly history of the transatlantic slave trade. Part of the reason for going that direction is we have very detailed historical records for this. It was a fairly permanent geographic separation, and we had accessible data for African Americans and Africans. It's from a data perspective made for a very good test case of, can we see the genetic echo of the transatlantic slave trade where African and African Americans separate on the family tree? Can we date that better with a young Earth timescale or with an old Earth timescale? And you can see we presented some of our initial results at a Creation Research Society meeting. We're about to write up our results, and one of the things that had bothered me, because this was the only data we had at that point, no father-son mutation rates, no human population growth history. One of the things that I did as I was thinking through our paper, as we're writing it up, well, what would the critics say? Let me be my own worst critic. What are some of the shortcomings in this paper? And one of the things that bothered me was, well, what do you do with the rest of the tree? All we've done is look at this tiny little segment that's just the African and African American branches. There's all this other part of the Y chromosome mitochondrial DNA-based family tree. And people are going to say, well, how do you explain that? That's what led to what I've narrated for you so far, the 2019 papers. And in fact, one of the linchpins in getting to the conclusions that I did was the Native American lineages. One of the things I learned in graduate school is you take something that you know and you beat it to death until it yields all of its secrets. So I said, we know there's been a massive population collapse. Where is the smoking gun in our DNA? And it turns out the Native American lineage, known as haplogroup Q, that branch, contains it loud and clear. What do you see over and over again in these Q branches? Flatlining, 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 flatlining. And then late in history, branches increasing. This is a diagram from the book 2022, but in 2020 is when I published, based on a different data set, the initial results. The graph of the history of the Native Americans in this branch Q rises, flatlines, recovers light. So a regional test of my predictions published in 2020, I confirmed this in 2022. So one of the additional predictions this finding makes is that additional data sets should confirm the same thing. Got another data set of Native Americans from the Bergstrom et al. Paper, if you want the technical background. I think I have it referenced in the book. There's another Q branch set of Native Americans, same sort of thing. Flatlining, centered around 1492. It flatlines before then again because you've got whole villages gone. So the early third cousins, fifth cousins, those branches are gone from the lineages of the survivors because they don't leave descendants. Anyway, you see the same sort of thing. One last confirmation on a regional scale, perhaps one of the true chronological events that's a true testable prediction. I've been thinking about Native Americans since 2017-18 when we were thinking about the transatlantic slave trade. One of the data sets that I didn't have any access to, 17, 18, 19, finally in 2020, after I'd published the global population growth curve, Bergstrom et al. In Science published a data set that included North Africans. I had no full Y chromosome sequences from North Africans up to this point. They included Mozabites, who are from Algeria. The population history of Algeria from archaeology and historical records is this. The solid black line represents the dynamic history of human population growth in Algeria. The flat line here, the dashed gray line represents the sort of the minimum of all this. You can see in my book sort of the longer justification, also the derivation of some of this in that 2019 paper looking at global history. My point is, I'm going to use the dashed line. You have to look up other stuff previous for the justification for why, because I don't have time for it now. I'm going to compare it to this new genetic data set. So we know from history and historical and from archaeology, here's how population history looked in Algeria. Does the population history of the Mozabites, based on their DNA and a young Earth framework, accurately recapitulate this? This axis represents the history, and the yellow and the gold represents the history from DNA, Y chromosome. 19 men. That's this yellow or this gold curve. The dashed black line represents what I had in gray in the previous slide. 19 men, the DNA from 19 men in the young Earth timescale can recapitulate almost 2,000 years of Northern history. When I saw this, I thought, okay, I think we're on the right track. This model actually makes predictions, because again, I didn't have it. So then the data set comes out. I thought, well, I guess we should probably look at this, test the predictions. I don't know how this is going to turn out. And when it turned out this way, I thought, okay, sigh of relief. I think this model is working. Yep, we've excluded ancient DNA, but I've got experimental evidence for why. And I continue to use the DNA from living men, and it keeps working. So the global results predicted regional matches. These regional matches have turned out to be true. I didn't even have time to go through some of the other, through some of the Middle Eastern lineages. I talk about it in the book where you see additional matches. The bigger picture point again is, how do we know that the conclusions in this book traced are true? The gold standard of science is testable predictions. And I'm narrating for you a sequence of predictions, discoveries, more predictions, discoveries that keep working. And this, for me personally, is what has given me confidence that we're on the right track and why I make the conclusions that I do in this book. The story's still not done. There's more predictions that flow from this. There's been a lot that's happened the last few years. We're now at, so I give you 2019 and then the third paper 2020. There's still more that happens between 2020 and this book being released in 2022. So stay tuned for the next episode. More predictions, fulfillments. Again, there's the larger question of the origin of species from a similar prediction fulfillment analysis in this book, Replacing Darwin. Cliff Notes version of this, Replacing Darwin Made Simple. You can find me on social media, Facebook, other places, which I've set up for the purposes of interaction. I hope you'll join us again in subsequent episodes where we'll continue to follow this story and then subsequently talk about more of the backstory.
Well, again, that is so wonderful to see and to hear that good science being done. And it makes sense to do good science within the biblical worldview, because science makes sense within the biblical worldview. And then we're seeing those predictions being fulfilled. And then also, I love just the clear statement you're making. Hey, there's more to come. There's more stuff we have to do. There's more work to be done, more science to be done, more research to be done. And I know you've said this a couple of times in the past, but I'm sure those who are watching, if you want to jump in and be a part of this and be a part of the research, I know Dr. Nathaniel Jeanson would love to have you be a part of that, fill in some of these gaps. We'll keep expanding this research for the glory of God and really showing how the Bible is confirmed again and again by real science. So be sure to get plugged in. Go to the links that he showed earlier to maybe be a part of the research, a part of this process, a part of this gold standard of science, if you will. So excited to watch all this just be fleshed out as we go through these different videos. Looking forward to the next videos to come. You guys be back then. Take care.